Welcome to Wave Club, everybody. The plan for Wave Club is that this is a monthly meeting run by myself with Irfan Zabeh, who's a student in my lab, Uma Mohan, you can see in the corner there, and Bart Ermentrout. And the plan for Wave Club, as many of you know, is this is going to be a monthly, I want this to be as informal and fun as possible, meeting to talk about traveling waves in the brain. And traveling waves in the brain in all ways are the topic of Wave Club as far as animal recordings, human recordings, behavioral studies related to things like traveling waves. The goal of this is to have it be a really inclusive and open sort of group to talk about all different aspects of this. I think that traveling waves are an exciting new area, and the more attention we can bring to this, and use Zoom to kind of bring different people together who are interested in this topic, is really the goal of this meeting. So today at our second Wave Club, Zach Davis from the University of Utah is going to be our presenter. I'm going to give an introduction to Zach in a moment. Next month, we have Laura Duget on June 4th. It's going to the standard time is going to be Tuesdays at noon. So Laura's going to be a presenter, and then plan for this going forward is to have different people each month. And the plan for each Wave Club meeting is going to be summarized here. We aim to have a 40 or 45-minute presentation, to keep things focused, what I'd like to do is to have the questions be saved for the end. And the most helpful possible thing is for any of the questions that you have for the presenter to be used in the Q&A feature. You should see on your screen, we're using Zoom's webinar format, and the goal is to have this Q&A feature where we can keep track of all the questions that pop up. And then Uma and Irfan were going to help me keep track of which questions are we're going to ask at each point in that 15 or 20-minute period at the end of the talk. And then what we'd like to do is bring the whoever asked the question up on stage virtually to ask their questions and have a little bit of a conversation and back and forth. So that's the plan. If you are not in a place with good internet, or you're a little shy, you would prefer that we ask the questions directly, that's fine too. Just let us know via the chat. But it's sometimes fun to have a little bit of a back and forth if we can. Hopefully many of you are on the email list, which is great. But the plan is to have different speakers each month, and self-nominations for speakers or non-self-nominations for speakers are all welcome as well. So thank you all for coming. Our presenter today is Zach Davis. Zach is a new assistant professor at the University of Utah. He has undergraduate training in psychology at UC Berkeley. Then he became a neuroscientist at the UC Davis, and he moved to the Salk Institute following his PhD at Davis to work with John Reynolds. And then as part of his work during his postdoc, he published some of the most influential papers on traveling waves that I'm aware of. And that's one of the reasons why I'm happy to have him here, because his work is really great. He's done both computational modeling as well as empirical studies of traveling waves. In particular, his 2020 paper that was published in a journal that you might have heard of called Nature. All that led into his opening his lab this year at the University of Utah, where he is right now, and he's been for 4 months or so. So I'm going to stop my screen sharing and turn over the screen to Zach. And Zach's going to present for 40 or 45 minutes or so. And we'll go from there. Thank you all for coming, and thanks for your attention. It's nice to see so many people here. I'm really excited.
All right, well, great. Thank you for that introduction. And thank you everyone for being here. It's a pleasure to present our work. But yeah, my name's Zach Davis. And so Josh did a very nice job of explaining sort of my academic background, but I thought I'd share a little bit more about my personal background. So some of you might be familiar with my work, but don't really know about me. And I think this club is a good environment to kind of have a more informal introduction. So this is a picture of the Moran Eye Center at the University of Utah, where my lab is currently located. Actually, it's in construction, but our histology lab will be up here on the 6th floor. And then across the street is where the animal colony is. And that's where we'll be doing our electrophysiology recordings. But before I really got into neuroscience, after I graduated from undergraduate, I worked in a private cognitive neuroscience lab in San Francisco. And before I was studying brain traveling waves, I was studying just good old-fashioned brain EEG waves. And so I don't know how many of you are familiar with the cocktail party effect, but we tried to study that in real life by wearing wireless EEG headsets and having a cocktail party. The actual scientific motivation was not very well defined, but I think it was just a good excuse to use up some extra money that was lying around from some grant. But this was actually published towards measuring brain function on groups of people in the real world. Yeah, so you can have fun too. When I was in graduate school, I was also on the UC Davis cycling team. So here's a picture of me questioning some of my life choices. And then my other claim to fame is that my dad was a video game programmer, and you might have heard of some of his games, but the claim to fame is Qbert. So my dad, Warren Davis, was the programmer for Qbert. So that's a fun bit of trivia if anyone out there is interested. And then the last thing, I have two dogs. Bogey on the left, he's a Vizsla. Mazzy on the right is a Greyhound. They're the first and third fastest dogs by breed. Although the funny thing is, Mazzy the Greyhound, she would rather not ever run. She just likes to sleep all day. Bogey, he loves to run. So, all right, let's get started. So the question that the lab that I'm currently building is interested essentially in what are the neural mechanisms that underlie visual perception. And here's a picture of the human brain, but the model that I study this in is in the marmoset monkey. There's lots of reasons why the marmoset's an appealing model for studying visual perception, but the one that's most relevant for us is people who are interested in spatiotemporal activity patterns in the brain, like traveling waves, is that the marmoset has a smooth cortical surface. And so this allows us to be able to interrogate large-scale spatiotemporal dynamics and how they interact without losing access to tissue into a sulcus. And so this gives us a means to be able to ask direct questions about how these types of activity patterns are coordinated within or across cortical areas in sort of a tractable fashion. And so the way in which we think about this thing traditionally is, there's some visual information out in the world. That information is captured by the retina, and that's then transferred to the thalamus and into the cortex. And there, through a series of hierarchical filtering, features are extracted from that visual information, and we construct this percept of what's happening in the external world. But the problem with that view, and I think if you were here last month for Earl Miller's talk, he did a really wonderful job of explaining how these types of frameworks don't really capture the emergent properties of things like visual perception in this context. And I'm not going to sort of review the complexity there, but instead I'm just going to offer up this as an example. So this video is an example of an ambiguous or a percept that can be bistable. So there's two equally acceptable interpretations of this visual stimulus. You can see it as a rotating cylinder rotating from left to right, with the foreground going from left to right, the background going from right to left. But the exact same input could be equally valid interpretation is that it's rotating in the opposite direction. And if you continue to stare at it, you might actually see it spontaneously transition from one direction to the other. And so this is a stimulus that's been highly studied in visual neuroscience. Christine Krug, Andrew Parker, Bruce Cummings have all looked at how this perceptual phenomenon is a consequence of the activity of cortical populations in the brain. And it's hard to imagine how this perceptual experience could be the product of a series of feed-forward filtering. Instead, it seems much more likely that there's some percept that is being imposed on the visual information that we're collecting. And so understanding the mechanisms through which the brain can do that is an interesting question. And I think understanding traveling wave dynamics in the brain might give us some insight into how this might occur. And then here, I'm just showing this image because I think now as the technology is allowing us to look at recordings from multiple cortical areas at the same time, calcium imaging, wide field imaging, neuropixels probes, Utah rays, et cetera, we now have the ability to ask pointed questions about how activity within cortical areas, but also across cortical areas, are coordinated to serve this inference of perception and how we're interpreting the visual information that's being collected from the retina. And I think this applies to other sensory systems and other neural systems in general. It's just that for me, the visual system creates a very tractable way of asking these questions. And so today in my talk, I'm going to focus on this idea of variability in percepts, the ability for multiple perceptual events to occur given the same visual input, is going to be linked to how that information is being interpreted by the activity, how it's being represented by the activity of neural populations, and how variability in that is going to be linked to perceptual variability. And so I'm going to provide some evidence that the variability in neural activity that we think underlies this perceptual variability can be attributed in part to fluctuations in activity that travel as waves across cortical surfaces. And then I'm going to give a mechanistic explanation for how we view traveling waves occur and how they have their effect on computations in the brain. And that is through synaptic fluctuations that are caused by spikes traversing the organization of anatomical connectivity within cortical networks. And so last month, Earl Miller gave a really wonderful talk, and he posited that traveling waves could be the consequence of aphaptic signaling. So actual changes in electrical fields impacting the activity of neural populations. And so I want to offer what I'll be presenting as an alternative mechanistic interpretation. I don't want to deny the possibility of aphapsis as a mechanism or deny that neurons can be sensitive to changes in electrical field. But the argument that I'm going to make is that we can describe much of the effects of waves as a consequence of spiking in synapses mechanistically. And while aphapsis could play a role, until we find ourselves exhausting the descriptive ability of these mechanisms, I think we should reserve other mechanisms until we're forced to fall back onto them. And for the last part, I'll be presenting some unpublished work looking at how the specific patterns of waves we believe reflect uniquely correlated fluctuations amongst neurons that are sharing tuning preferences through their preferential connectivity. And so we believe that these are a reflection of functional ensembles in the network dynamics, and they're interacting from moment to moment, and how that interplay unfolds has an impact on sensory processing and perceptual sensitivity in a feature-specific domain. And I'd like to highlight that this work is not just my own, but it's the product of a team of really highly skilled people, starting with John Reynolds, my postdoctoral advisor, but also Lyle Mueller, who's been really an essential collaborator in getting this work across, especially on the computational modeling and the sort of theoretical side that underpins what we think is going on in the brain. And so I met him when he was at the Salk working with Terry Sędziowski. And then I've gotten to work with Lyle as he's been a professor at Western University, working with his lab members, Gabriel, Alex, and Christopher, who've been helping out on all of the modeling aspects of this work. And then Julio Martinez helped with some of the early experimental design for the studies. And then Charlie and Theo were instrumental in the foundational architecture for the modeling framework that I'll be presenting later. Okay, but so starting with this idea about perceptual variability and neuronal variability. So I gave you this example of this rotating cylinder, this bistable stimulus, and this is kind of a complex thing. There's a lot going on. So to sort of start with the simplest example of this that I can think of, at least, is the concept of the threshold detection test. So here, the question is, I'm showing you something, you saw it or you didn't, why is that? And so this is something that we can easily train marmoset monkeys to do. They do it quite naturally. And so the way in which we do it, and the task that was done for this data, was we start by showing a head-fixed marmoset. We're doing eye tracking. We show them a picture of a face. That's something that they just naturally like to look at, that draws their gaze. It turns into a fixation point, and they've been trained to hold fixation on this point, awaiting the appearance of a target somewhere. They don't know exactly where it's going to appear. They don't know exactly when it's going to appear. They just know that if it appears and they see it, and they indicate that they've seen it by making an eye movement towards it, they get a juice reward. And so they'll do hundreds and hundreds of these trials a day. And this allows us to vary the luminance contrast or the salience of the target, which here in this example, it's a drifting Gabor stimulus. And it's drifting in one of several directions, but we can take all the times when the stimulus is identical and we can change the contrast. And what we find is that when it's really easy to see, the monkey is very accurate in reporting that it's there. When it's very difficult or impossible to see, then the monkey is at chance or unable to report it. But what we can do is we can find a value at which the monkey detects it about half the time. And so this psychometric function on the bottom left, this is data from 2 monkeys, and they actually had roughly similar thresholds for about 50% detection rates. And what this means is that on any given trial, the stimulus is the same. It's an identical visual input, and yet sometimes the monkey saw it, sometimes the monkey didn't. Why was it that that occurs on a trial by trial basis? And so here we're recording from neurons in area MT, which is an area that's sensitive to motion, which is useful because our stimulus is a drifting Gabor. And here I'm just going to highlight that we're recording from, in this example, an individual neuron, but in actuality, we're recording from Utah rays that we've chronically implanted. And the nice thing about MT and the marmoset is that it's a tractable size. We can record from the entire visual field within the space of a recording chamber or an implanted recording array like a Utah ray. But for now, we're just going to focus on a single neuron. We can find its receptive field from reverse correlation receptive field mapping, a standard technique. And then we can ask when the target is presented into the receptive field of the neuron that we're reporting from, what is the response? And so here's an example where that's the time of the red line is when the stimulus was presented. And then the vertical tick marks here are the spikes that was reported assigned to this unit that we've isolated. And so this is another time where we present the same target, another time, another time, another time. And the point here is that despite the visual input being the same, because we're controlling the gaze of the monkey, we're controlling the properties of the visual input, this variability is not due to the stimulus. It has to be due to something intrinsic to the environment that this neuron is operating in. And so if we look across many, many presentations of this target, we can see that there's a lot of variability, but we can sort these based on whether they were times when the monkey saw the target on the bottom or failed to see the target on the top. And so what I'll hope you appreciate is that there's a direct correlation between the magnitude of the evoked response, how many spikes this neuron fired when the target was presented, and whether or not the monkey saw the target. So the question becomes, what was going on each one of these instances that produced a stronger or weaker response to the exact same input? Now, we know that neurons in the cortex, they receive strong driving input from visual sources. So in the example of V1, therefore neurons get strong driving input from the thalamus. But this only accounts for a small fraction of the total synaptic inputs that a neuron might receive. In fact, up to 80% of the synapses that are connecting to a neuron are coming from recurrent connections locally within the cortical network. And this interaction creates an environment where you're having constant synaptic fluctuations that are ongoing independent of the specific feed-forward input that's arriving. So that input is arriving into an environment that it can be either more or less depolarized is the idea. And so what we'd like to do is to record the spiking activity and the synaptic potentials of every cell in a cortical population to be able to understand how this is all playing out. But that's just not a practical solution with the given technology. So instead, we can take as a proxy for the synaptic activity in the local population in aggregate, the local field potential, which fluctuates with changes in excitation and inhibition as these recurrent synapses are being activated in the local population. But the sources of those synapses are coming, especially in the superficial layers and also in the deep layers of cortex, from horizontal fibers that send processes that extend across millimeters of cortex. And so if we really want to appreciate how all of these synaptic fluctuations are interacting dynamically across a cortical area, then we need to record from the entire cortical area to at least have a hope of being able to understand how these things are unfolding in space and time. And so for that, we use these chronically implanted Utah arrays. There's lots of other ways that you can do this. But the take home is, if you look at the LFP fluctuations that are occurring across all of these channels, across the cortical areas, they have this structure of a traveling wave. And so here I'm preaching to the choir. We all are aware that these dynamics occur and that we can see them. And so the question becomes, how much do these waves explain about moment-to-moment fluctuations in the intrinsic excitability of the underlying cortical populations? And so because in this example, we're looking at a situation where the monkey's fixating on the screen, there's no visual input, we're controlling for eye movements, there's no other source that we can attribute these things exogenously. So we call these intrinsic traveling waves because we feel that they emerge from intrinsic fluctuations in synaptic activity driven by other sources, other cortical areas, other just local recurrence. We're agnostic to that. We just say it's coming from within the brain. Okay, so how do we relate these wave dynamics to the variability that we believe is contributing to fluctuations in perceptual sensitivity in the example of this detection task. So the way in which that we do it, and this is an algorithm that was developed by Lyle Mueller, is we look for divergence in phase from a putative source in our electrode recordings as a function of distance. And so there's a lot of advantages to looking for waves in data in this way, especially given the sort of noisy nature of multi-channel electrophysiology recordings. But what this allows us to do is create a statistical test where we can say how correlated are the offsets in phase from this putative source point as a function of distance, and how does that compare to a spatial shuffling? How much spatiotemporal structure would we just expect to see by chance by virtue of the relationship of the activity between the electrodes? And by performing this permutation test, we can create a null distribution of correlations of phase with distance, or rho phi d here, and then we can identify periods where we can say statistically that there's more spatiotemporal structure here than we would expect by chance. And there's lots of advantages to this technique that I won't go into right now, but the key thing is that this is a phase-based measure. And so the question becomes the phase of what? And this becomes challenging in recordings of the visual cortex because activity there, especially here during awake fixation of a blank screen, but also under other sensory conditions, is only transiently oscillatory. So unlike areas like the motor cortex, where there are these prominent beta waves that have been leveraged to show evidence of traveling waves and their effect there, here it's hard to identify a specific narrowband frequency that we could filter in that would describe a large fraction of the data here. So this here is an example of a single LFP recording from one of the electrodes in our experiment. Here it's filtered from 5 to 100 hertz to kind of show the breadth of frequency content. We use a 5 hertz high pass filter because we want to eliminate a lot of the large amplitude, low frequency fluctuations that are in our data because, one, we attribute these things mechanistically to arousal signals, but two, it makes it difficult to dissociate those large amplitude fluctuations that are occurring on a slower time scale than the wave dynamics that we're trying to extract. So we throw out that data. There's interesting stuff, I believe, happening in those lower frequencies, but for the purposes of this, we're excluding those from our analyses. So here is an example of the power spectral density of this trace here in black. In gray are a bunch of other traces from other trials, and then the red dashed line is the average across all of this data from this recording session. And so what I just want you to appreciate here is, while there is a peak in the power spectrum, that only describes a subset of the time in which that frequency is showing pronounced power. There are other times where the spectral content is not of that frequency because there's power in other frequency bands. And across trials, that peak is not even at the same frequency, it's moving all over the place. So how are we going to address this issue? So this is a chunk of recording, and this is a nice example because you can appreciate that from moment to moment, the frequency content of this signal is changing. And so what happens is if we use, let's say, a classic filter for visual cortex, filtering data in this alpha band, this 8 to 13 hertz. Well, we might be able to describe the phase of this signal accurately at certain times, but at other times we're going to be doing a very poor job of describing the underlying signal that was going on in the brain. We can pick maybe a filter band that's more suited for the peak in the power spectrum, here, this beta signal, and it does a very good job most of the time, as you might expect, because a lot of the time, by virtue that there's a peak in the power spectrum, we know that there's a lot of frequency content in the signal at this frequency. But at other times, we're still going to be missing the mark here. Here's an example where the filter signal is going up, but the actual real data is going down. Here's another example. The signal is going up, the real data is going, the real data is going up, the signal is going down. So we're missing out on a lot of our data. Either we're poorly identifying what the actual data was, or we're imposing something on here that wasn't actually happening. So what we find, it's much better to take a more frequency agnostic approach and say, rather than trying to ask specific questions about the frequency domain, say, I just want to know what the signal was doing at each moment in time as it drifted in frequency content from moment to moment. And to preserve that information, I'm going to use a wider band frequency, a wider band pass. And now when I take the phase of this filtered signal here from 5 to 40 hertz, I have what we call a measure of generalized phase. So this is a phase that is not of an oscillation, but of the underlying signal in where most of the frequency content is as it's drifting in frequency from moment to moment. And so we can now ask a question: is this representation of the data better or more useful than a narrowband interpretation? So here's an example of us doing this process of identifying the generalized phase of this, we call it wideband subset of the frequency content of the signal. And based on sort of ideas about what the underlying LFP is reflecting, we can say, well, this is fluctuations in EI balance and synaptic activity. We're going to call the troughs here a depolarized state where the EI balance is shifted towards E. We're going to call the peaks a hyperpolarized state. EI balance is shifted towards I. And we can say, how well does this capture the spontaneous variability and spiking activity in this framework? And so we can see here that it does actually a very good job. If we look in the depolarized state, so here we're looking at the probability of a spontaneous spike occurring as a function of the phase of this, this wideband signal, we see that spikes are more likely during the depolarized state and spikes are less likely during the hyperpolarized state. So we're doing a pretty good job of recovering something fundamental about the synaptic activity that's affecting spike probability in neurons. Well, how does that compare to other measures? Well, we can filter in a bunch of different classic frequency bands like theta, alpha, beta, gamma, and we can ask this empirical question: what frequency band does best? And here what we see, each dot here is the magnitude of the spike phase coupling relationship to the LFP averaged across an entire recording. And so each dot here is a single recording. And so this is across many recordings from within 2 monkeys. And what we can appreciate is that you're able to describe some of the spike phase relationship, but it's always impoverished compared to this wideband representation. And if it was the case that spikes were uniquely coupled to some underlying frequency, it would be reversed. Then it would be the case that filtering actually would help you. And so this is an argument that spikes are not actually coupled to a specific oscillatory frequency, but they're coupled to the broadband spectrum as it's changing in frequency content from moment to moment. Now you might say, well, maybe it's the case that spikes are coupled to alpha when alpha is present, spikes are coupled to beta when beta is present. Well, we can test that by saying, well, we'll apply these filters, but we'll only include data where the attenuation of the signal is below some threshold. So as long as we have maintained much of the raw signal in there after filtering, then we'll include it. But even then, we find that with these narrowband filters, we're still losing out on the maximum amount of predictive power we have for assigning spikes to these fluctuations in synaptic balance. And the other consequences, even though we're doing better, we're still leaving out much of our data. So here, these percentages are describing how much of the data pass our -5 decibel signal to noise ratio threshold. And so here, 60% for alpha, for theta, 46, etc. But so even better, these filter bands are somewhat artificial. We can just parametrically say, what is the best bandpass filter to just sort of coarsely apply to our data? And so what we're looking at here is every pixel is filtering the data in a different bandpass filter. We're doing it from a range of low band, so the high pass from 1 to 50 hertz, and then here the low pass, the upper band from 5 to 100 hertz. And the white pixels are band passes that don't make any sense. These are, you can't filter from 10 to 5, you get it. So what I'd like to draw your attention to is that all of the points here along this boundary are very, 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 very narrow band. So 2 hertz band pass minimum, I think, was what I set for the threshold criteria to be included. And what you'll see is that these filters do a very poor job of capturing the spike phase relationship. As you widen the filter band to broader and broader, it gets hotter and hotter. So that means as you include both lower and upper frequencies in your bandpass, you do a better job of capturing the relationship between spiking and LFP phase. And I should point out that this is after doing a technique to despike the LFP to delete the contribution of the spike itself from the raw data developed by Chris Pack's lab. And whether that's the best way to do it, I don't know, but it allows us to at least say that this isn't due to the presence of the spikes themselves. And there's other checks we can make to ensure that. But the point of this sort of the point of this whole discussion is really to state that if we're going to look at traveling waves in the cortex, we want to be sensitive to variability in frequency and time, but also variability in frequency across electrodes. And if we take a narrowband approach, we're imposing a structure on the data that might not be there. And by taking this wider band approach, we're allowing the data to sort of breathe in its own way and be sensitive to those changes as they occur. Okay, so now that's all set up. We have our method of detecting waves. And now we can ask this hypothesis, when these waves are occurring, do they reflect shifts in excitability that impacts both the gain of the evoked response and the sensitivity of the monkey performing the test? And the sort of conceptual framework for this hypothesis is that we have some spikes coming in from the source, the visual input. And those are arriving into an environment that can be more or less depolarized. And so if it's arriving into a relatively hyperpolarized state, you're going to get a weaker evoked response. But as these waves traverse the area, they can align more depolarized states with that same sensory input and then give you a bigger bang for your buck. And that are in the framework is that more spikes means higher likelihood that those are going to pass some threshold for detection, and it will breach this threshold for conscious perception, which is a phrase that I'm taking from Peter Rolfson's lab. Okay, so I'm not going to go through all the methodology for how we did this analysis. I just want to state the results, which is if we tried to ask whether or not a more depolarized or more hyperpolarized wave state is aligned with the onset of the evoked response, we got exactly what we would expect. A more depolarized state produced a stronger evoked response, shown here in green, and a more hyperpolarized state produced a weaker evoked response, shown here in purple. And this had an impact on the monkey's ability to detect the target. If the target arrived in a more depolarized wave state, the monkey was about 50% more likely to detect the target. If it was in a hyperpolarized wave state, the monkey was about 50% less likely to detect the target. So for a task where on average, the monkey was seeing the target about half the time, this was, 65% to 35% change in detection probability. Now, this idea about sort of pre-stimulus factors predicting perceptual performance is not a new idea. And so we wanted to ask, how well do these traveling wave states compare to other measures that have been shown to be predictive of performance? And so we set up a generalized linear model to try to predict hit or miss on each trial based on wave phase, but other predictors like pupil diameter, which is a measure of arousal, and even the target evoked firing rate, which at the very beginning was the thing I said was so instructive as to whether or not the monkey was going to see the target. It turns out that the wave phase is actually more predictive than the spike rate for an individual neuron itself. And I have a lot of ideas for why this might be the case, which we can discuss. But these are the results that we found. And then these other measures, pre-target alpha power and pre-target firing rate, could be an index of attentional state. This wasn't an attentional task, so it might not be surprising that they weren't very predictive. But we threw these in here as well because there have been some reports in the literature. Okay, so I've laid out a framework for conceptually for how these waves can impact gain and perceptual sensitivity. But what are these mechanistically, or at least what we think are they mechanistically? And can we explain their effects in the context of spikes and synapses? So one idea comes from the observation that the waves that we see in the cortex,
about 10 minutes, if you don't mind.
Oh, thank you.
Sorry to interrupt. Go ahead. Thanks.
No, please. So the waves we see in the cortex tend to travel at the conduction velocity of the unmyelinated horizontal fibers within a cortical area. So this is a figure from Ian Nahaus and Matteo Carandini's work, and we see very similar things. So this gives us the idea that maybe the waves that we see in cortex are the product of these things traversing through these horizontal fibers. So to test this mechanistically, we set up a spiking network model where we instantiated this connectivity principle from the horizontal fiber network into the structure of the model to see if it produced traveling waves and if they could explain the effects we see in vivo. I'm not going to go into the nuts and bolts of the model. The key components here are just that the connections are drawn from a Gaussian, and this is to emulate the property that adjacent neurons in cortex are more likely to be connected, distant neurons in cortex are less likely to be connected, but that tends to fall off with some spatial term. And the second component is that once a neuron spikes in this model, its synaptic partners won't receive their signal, the synapses won't transfer current until after a delay occurs that's proportional to the distance and to emulate the idea of a spike traversing that fiber. So this principle of spatial connectivity and then distance-dependent delays form the framework that differentiates this model. The last thing is that this model is very large scale. It has hundreds of thousands to million neurons with thousands of connections each. That turns out to be important, but I'm going to skip that for now. But the take home is if we look at spiking activity in this model, it can produce waves, traveling waves of spiking activity. So we call these sparse waves. It's a little bit deceptive showing this figure because here we're looking at 80,000 neurons, and they don't all fit on the screen. So the spike size themselves are actually larger proportionally than the width of the raster. So just to kind of highlight this, if we take this little strip here, this strip actually contains like 300 neurons. And what I want to point out here is that the activity here is locally asynchronous irregular. So it looks like cortical activity if you just had a single tungsten electrode of the cortex. But as you zoom out, as you appreciate this on the scale of all of the neurons in the network, you can see embedded in that these subtle fluctuations that travel as waves. And so that's why we call these sparse waves. And there's some important distinctions there that, again, in the interest of time, I won't get into, but happy to discuss. But so now in the model, we do have access to every synapse and every spike and every neuron. But to compare this to our recordings where we don't, we wanted to make a proxy for the local field potential. And so here we're using a model developed by Bizzoni and the Einval lab, looking where they identified that one way you could describe LFP fluctuations as the consequence of fluctuations in excitatory and inhibitory synaptic activity as they fluctuate in their balance from moment to moment. And so we use that to apply a model of the LFP to pools of synaptic activity in the network, and then produce something that qualitatively looks very similar to the kinds of LFPs you might record in vivo. But importantly, this is not, there's no mechanism of the LFP in this model. The LFP is acausal, but it's a reflection of what we think is the real mechanism, which is the coordinated activity of synapses because of the structure of the anatomical connectivity. And so this is sufficient to replicate the relationship between spiking and the phase of the LFP as we see in our cortical recordings. And also it produces wave dynamics. So here each pixel is one of these LFP pools. And what we're looking at is the amplitude of that as it unfolds across the sort of spatial extent of the network. And then the last thing the model can do is it can replicate this phenomenon of gain-dependent modulations in evoked activity. So here we're providing an input to the model as if it was a visual stimulus. And depending on if it was in the more depolarized or hyperpolarized phase of these dynamics, you get this gain modulation that sort of emulates the gain modulation we saw in vivo in our V12 ray recordings. Okay, so for the last 5 minutes or so, I want to present some new work that we're in the process of getting published right now that sets up a question that is sort of in the same line about testing this hypothesis that these wave dynamics are emerging from horizontal fiber connectivity. So one feature of horizontal fiber networks in the visual cortex, but in other cortical areas, is that they form clustered connections with light-tuned neurons. So a classic example of this, if you think about the orientation pinwheel tuning of primary visual cortex, if one were to inject a tracer into this sort of blue-green patch on the top left corner and ask, where are all of these neurons sending their processes to and making connections, you see this patchy connectivity pattern that overlays with regions in the cortex that have similar tuning preferences. And so the probability of two neurons making connection are proportional to their difference in tuning preference. And so we thought, well, if we impose this kind of tuning connectivity and selectivity in our model, does that create any testable predictions that we can look at in our data? And so we created, here each pixel is the tuning preference of a neuron in our network model. And the colors here correspond to tuning preferences for different directions of motion. And then we can tile these pinwheels across the spatial extent of our network and add to our connectivity rule, instead of just the local Gaussian, which is this hotspot here, a marginal connection probability that is dependent upon the tuning distance once you get outside of that Gaussian. And so the product of these two connectivity rules is that you have here, for this example, every black dot here is a synaptic connection between one neuron located here in this little green patch. So you have sort of tuning independent connectivity based on the Gaussian, but then long range clustered connections that tile with the locations of tuning of light tuned neurons throughout the network. And so one of the consequences of this sort of diversification of the connectivity is that it produces more diversity in the kinds of patterns we see in the network dynamics. I'm not going to go into too much detail about how we do this, but I'll just say we're looking at phase patterns that are produced by the LFP pools that we're modeling in the network, and we're asking how similar or how different are these patterns from one another. So here's an example of two phase patterns on the top row. This is not showing up. That are highly similar, and the below that are two patterns that are distinct. And the point is that we can do some clustering analyses to group patterns that are more similar than you'd expect by chance, given the dynamics of the data. And we call these grouping of like phase patterns motifs. And so one question is, do these motifs have any functional, are they reflecting anything functional about the network? And the answer is yes. They tend to reflect shared tuning dynamics or spontaneous fluctuations in shared tuning dynamics. So here, these are three examples of motifs. The top row are one pattern. The second row is a repetition of a similar pattern. And if we group all the neurons in our network based on their tuning preference and ask, what is the spontaneous activity of that population when we see repetitions of motif 1 versus motif 2 or motif 3, we find that they're tuned. So that means that the motif, when motif 1 is occurring, for example, neurons that like down to the right tend to have more spontaneous activity. Neurons that like up and to the right have less. And so this sets up two predictions we can test in our data. Are there identifiable motifs that repeat in the sort of fine scale dynamics of the waves that we identify? And do those repeating patterns reflect coordinated fluctuations among like-tuned neurons? Well, luckily, from our Utah array recordings, we have a diverse pool of like-tuned neurons that we can collapse across. And we can look in the phase patterns that we see during the waves that we observe. And so here's an example of one phase pattern that has in the central column 2 repeats, and then on the right are 2 phase patterns that wouldn't be classified as repetitions based on this measure of phase similarity as a function of space. And so just in the interest of time, I'm not going to go into this too much. Just the idea that when we look at all of the similarities in phase patterns, we get this phase similarity space, and we can kind of carve off patches of this that correspond to highly similar groups of patterns. And so these would be identified as motifs in our in vivo recordings. And we can ask, well, are these, as the model predicts, reflections of light-tuned populations? And the answer is yes. If we pool across the different neurons in our populations and ask during these motif patterns, is there a bias for one population over the other? The answer is yes. And we can do some controls to make sure or identify how much tuning we would expect to see just by virtue of intrinsic correlations amongst like-tuned neurons. And we find that we see more tuning than you'd expect by virtue of already known levels of correlation. And so to take it even a step beyond the model, we can then ask, well, does this actually have an impact on the evoked gain of targets and the perceptual sensitivity of the monkey? And the answer is yes. So if we look at whether or not a neuron fires a stronger or weaker response for a particular direction of motion for one of the targets in our task, if that motif was present, which we believe reflects depolarization amongst a subnetwork of like-tuned neurons, if that is the target that's preferred by that population, then the evoked response is actually higher. And if it's not, the evoked response is actually weaker. So separate from the sort of spatial dependence of these traveling waves, there's also layered on top of that a feature dependence, such that if a certain subnetwork is activated, then you get more spikes. And similarly, the monkey tended to do better when they got more spikes versus when they got less spikes. And so there's a similar effect on perceptual sensitivity for specific features of targets if one of these motifs was present at the time of the evoked response. But yeah, sorry to rush through there a little bit at the end. I just wanted to make sure I got that in, but also I really want to make sure we have time for discussion. So just to summarize everything, I presented some evidence in support of this idea that perception varies with the state of internal fluctuations in network activity. And these might be coordinated because of fluctuations that travel as waves across the cortical surface. But if we want to look at rhythmic fluctuations, at least in the visual cortex, it's difficult to do that if we just blanket apply a narrowband filter. We really want to capture the variability in the spectral content of these signals. Then we recommend using a broader representation. And then we've solved this issue by coming up with this idea of generalized phase. So we can explain mechanistically how waves emerge, at least within a cortical area, from the organization of horizontal fiber connections, and the properties and the effects that waves have on computation can be explained by virtue of the depolarizations that occur from spikes traversing those anatomical connections. And finally, these waves can reflect subnetworks that are coordinated because of the specific connectivity that's shared amongst like two neurons. And this might be a way in which both space and feature representations could be stabilized across multiple cortical areas by activating these subnetworks in a coordinated fashion to make sure all of these cortical areas are sort of on the same page when it comes to how they're influencing the sensory representations that are being built from the input coming in from the retina. But yeah, thank you much. Thank you all for your time. Thank you to all of our collaborators and John, my advisor, and all of our funding partners that have helped contribute to this work. And I would love to open up the discussion and see where we go from there.
Zach, that was awesome. Thank you so much. I'm not going to clap because that doesn't work well on Zoom, but accept my virtual clapping, please. I have a bunch of questions, but I'm going to let Uma handle the Q&A here. Uma, do you want to turn on your video camera and you can...
Hey, yes, I'm going to start with the first question from Mrugank. I'm going to make you a panelist really quickly, and you can ask in person. Or kind of in person.
I usually hate Zoom, but I love Zoom for Wave Club.
Hi, I'm audible. Okay, awesome. So that was a great talk. So just a quick question, like when you mentioned the intrinsic traveling waves, like is the origin of this some sort of like a feedback signal that's happening because of the task instructions, or is it like independent of it and it just happens naturally? Like, is it some sort of internal noise?
Yeah, so that's a really good question. So we don't ascribe an origin to these. They could come from feedback sources, they could come from other sources. We're a little agnostic to that at this point. Regarding whether or not they are specific to the task instructions, there could be a way in which task instructions affect the likelihood of certain wave patterns or wave timing from occurring, but the occurrence of these types of intrinsic traveling waves are not specific to this task. So we've looked under periods of fixation, we've looked under periods of freely viewing natural scene images where we take special care to control for eye movements, because eye movements can also cause traveling waves in visual areas. And we also have looked under periods of like blackout, like it's pure darkness. And so we see these types of wave dynamics under a whole host of sensory conditions and behavioral conditions. So their properties may change, but the phenomenon of waves is pretty ubiquitous across the experience.
I guess I had one more follow-up question, which I guess goes hand in hand with the question that was asked by someone in the chat, which was, is there a reason why you chose that particular frequency band? Like I can imagine you can fit to the LFP model if you just go all the way from 5 to 200 hertz, right? And in that case, what does phase even mean? Like phase is usually like it's the angle of a particular frequency band, but once you're taking multiple frequencies, it's like a circular sum of those phases. So what does it mean?
Yeah, so this is a really important technical question. So we in the paper used a band pass from 5 to 40 hertz, and there was some motivation for that. It wasn't just arbitrary. I think I already described why we did the 5 hertz. That helps solve some problems that makes the frequency or the phase measure more tractable, but also just conceptually, we attribute those signals to arousal that doesn't mean arousal is not interesting. That's just what we did. The low pass, the 40 hertz, that was done to mitigate concerns about the contribution of spike coupling, so spurious spike artifacts impacting the signal. And so we would just be reading out a sort of an echo of spiking as opposed to some underlying mechanistic relationship. So that was done to mitigate that. But in the later paper, where we did this more parametrically, we found that we could actually do roughly the same job from 50 hertz to 100 hertz. You gain marginally in that figure as you increase the upper band pass. Now, maybe that's because of the strategy we took with the despiking. We did look at it without the despiking. It didn't change the broadband very much. So I mean, it was sort of motivated by some technical concerns and motivated by some conceptual concerns, but that doesn't mean that's the best way to do it. And that was specific also to the data that we had in the visual cortex. As to your second question, which I think is the more important one regarding what does phase mean when you have a broad spectral signal, it sort of depends on what the signal content is. So if you had white noise and you filtered it across a broad range, and then try to ask what is the phase of that, I would agree with you. That's not a very, because it becomes with the phase of what? There's so many different frequencies that all have equal power, and they're all interacting in a complicated way. But when you look in neural data, you find that from moment to moment, the frequency content tends to be in a narrow range. The breadth of the power spectrum largely comes because we're looking across a long period of time. And so you can have power in lots of broad frequency ranges, but that doesn't mean all that frequency is there at any given moment. And so in the traces that I showed you, even though those have broad spectral content, you could by eye identify fluctuations that were flowing up and down. And if you want to describe whether something was moving towards a more positive or more negative voltage, you could get a pretty reasonable measure of phase that tracks with that really well. And so generalized phase is a technique that Lyle developed that basically solves some of those problems that you are feeling concerned with. So, for example, the easiest thing to think of is if you have some sort of low frequency thing that has a lot of amplitude, and then on top of that, you have a high frequency writing cycle that is small in amplitude. If you were to look at that in phase space, what you do is you have your phase clock, but it's doing all of these sort of loops, and that introduces periods of negative frequency. And so what the generalized phase algorithm does is it tries to say, look, we don't want to distort the shape of the signal that we're trying to recover, but also these high frequencies could be a problem. So we're going to throw out anytime we detect a negative frequency and interpolate over that. And that strategy is a way to try to restore some flow to the large amplitude underlying signal that we think is doing most of the driving when it comes to understanding the machinery of the brain underneath. Will this technique apply to other frequency decomposition things that are outside of neural systems? I don't know. But as far as it goes to try to understand what the brain is doing and how that relates to the underlying spiking activity, it appears to do a pretty good job of recovering signals that are more descriptive than other techniques. That doesn't mean it's the best way, but for us, it seems to be doing a good enough job that we're able to make some hay with it.
Awesome. Thanks.
Yeah, thank you.
Thank you. That was a really helpful explanation. Next, we have a question from Nico Hatsopoulos. Nico, you're muted.
Can you hear me now? Okay, great. Zach, great talk. Really enjoyed it.
Thank you.
I don't remember from your paper, was there any anisotropy in propagating directions that we like what we see in motor cortex?
Yeah, so it's a good question. When we looked at this, we just did a quick check to make sure we didn't see the same anisotropy we see with that Chris Pack saw with saccade-evoked waves. And we didn't see a strong bias in any particular direction. Now, we haven't exhausted the possibility that there could be some anisotropy for some types of wave patterns, but in general, when we looked for this, we didn't really see it.
Okay, great, thanks.
Yeah, but I will say we've looked recently at, let me see, where am I? Yeah, so recently we've been looking at some recordings that were published by Peter Rolfsma's lab. So this is from their 16 Utah array recording data. And so these are the 14 Utah arrays that were in V1. And looking in this data, it's a little bit different because now we're looking across a much, much larger cortical area across the visual field. And so we do see evidence that there might be a bias for some directions of motion here in the wave patterns. Here we see sort of more of a circular pattern. And this could be because the sampling resolution is different, or because here the monkey, I think, was in the dark. So it's hard to say whether or not there might be differences depending on state or condition that might drive anisotropy in visual areas in spontaneous activity. But yeah, in the recordings we did in the marmoset, we did not see strong evidence for it.
Great, and just to quickly follow up, I know you can't really know for sure, but what layer do you think you're targeting with these Utah arrays? Is it something that's interests me in motor cortex, whether there are different spatial temporal structure on different lamina?
Yeah, so this is a really great question. There's a lot going on in this figure. This is just this idea that we've done surface ECOG recordings, and we've looked to see whether or not there's different coupling across the layers using laminar probes to wave dynamics. So what we found is that superficial and deep layer neurons tend to show stronger effects than input layer neurons, but the effects can be seen across all layers. So because of that, it's hard to know for sure what layer our Utah arrays were in. We did some histology, but it wasn't perfect. So if I had to guess, I think that we were in the bottom of the layer, like the superficial layers. I don't think we were in layer 4, but I don't know for sure. But my intuition is that we were in the superficial layers. But that's all I can really say about it. Yeah, it would be, I think it's a really important question, a really interesting one to understand how these type of dynamics play out across the layers, which motivated this unpublished work that I'm showing right now. But yeah.
Were these 1 millimeter Utah arrays or 0.6?
They were 1.5 actually, but
They were 1.5. Okay.
I don't think they went in all the way. And the marmoset cortex is maybe 2.2 millimeters thick here. So it's hard to say, really, it is. The histology wasn't very compelling because the cortex got compressed histologically. It was kind of a mess to try to decipher what actually happened. But based on some other measures of spike field coupling across the layers and other properties that we observed, it makes me think we were in layer 2, 3. But I could be wrong. We could have been in layer 5, and you might predict that you'd see the exact same things. It's hard to say.
Thanks.
I just want to add, since I'm the MC and I can unmute myself whenever I want, I thought those were really interesting questions, and I thought it was really cool you were looking at Peter Rolfsma's data in that way, because I know of that data set, but I'd always wondered if there were traveling waves in there, and now I know the answer, so this is like 2 for 1.
Yeah, it's actually, if you look across the V1 and V4 recordings, you see waves in both V1 and V4, but there are some interesting coordinations going on. We're working on a manuscript right now as part of a collaboration with Adam Cohn and Andrew Parker, looking at these coordinations because the model, so we take our network models and we connect them, we see that waves become coordinated across cortical areas, so it allows us to test this prediction. So we've been looking at these Utah array recordings in V1 and V4 in macaques and finding that there are retinotopic relationships that are interesting. So that's a work in progress right now. But yeah, I think there's a lot of important insight that we can get from looking at these inter-area coordinations.
I think that's exciting, and that's exactly the kind of thing I would not know from only reading your papers. So this is fun for me. Nico, did you have any more questions? I kind of jumped in there. Okay. Uma, you want to take over?
Sure. Thank you, Nico. Our next question is from Elliot Smith.
Hey, how's it going? Can you hear me?
Hey, yes.
Cool. So yeah, awesome talk. My question was just about the model real quick, and sorry if I missed this, but
Yeah.
did you model both excitatory and inhibitory neurons and the connections between them, and then were the kernels the same for those connections?
Yeah. Yeah, this is a great question. Yeah, unfortunately, just in the interest of time, I didn't really break out the model. That's kind of an important part of the talk. So this is a great opportunity to kind of break that out. So the model is the neurons are leaky, integrate, and fire neurons. We have 80% excitatory, 20% inhibitory neurons. So they're just, those are the classes, excitatory and inhibitory. The synaptic activations are modeled as conductances. So depending on the membrane potentials of the neurons in the model, that's going to affect the amount of current that is passing when the channels are opening. And the topography of the connectivity for the Gaussian is the same for excitation and inhibition. So both neurons are modeled as making distance-dependent connections. And whether or not that's the best way to represent inhibitory neurons is, an open question. It just turns out that that's a useful way of making sure that everything is balanced without having to do any tinkering. But other groups like Brett Doran have looked at differing the scales, the spatial extent of excitatory and inhibitory connections, and finding interesting dynamics emerge from having differences in treating them differently. In the additional step where we added in the long-range tuning-dependent clustered connections, those were excitatory only. So only excitatory neurons would make long-range clustered connections. The inhibitory neurons only made local Gaussian connections. We did play with it both ways. It does affect things that we didn't really study fully, but it doesn't seem to be fundamental to the observation that you get these sort of unique motif patterns that are tuning dependent. Having inhibition the same as excitation in that model, if I recall, still worked roughly the same way. But we kind of abandoned that idea because we wanted to have a more better verisimilitude with what we think the actual source of the long-range horizontal connections are as being excitatory. So I don't want to commit to say that it doesn't like completely change things, but from when I was playing around with it, it didn't seem to make a huge difference, but we didn't really fully explore it.
Great, thank you. Next, we have a question from Li Hao Yan.
Right. Thank you, Professor Davis, for a great talk. I've read several of your papers, and it's great to see you actually talking about them. So I have two questions. The first question is, have you investigated the spike phase index for tasks other than the threshold detection? For example, if the marmoset is receiving a phased input, then we would imagine the local field potential to be correlated in phase to the input phase rather than to the spikes.
Yeah, so this is an interesting question. So all of the spike field relationship stuff that I showed was for spontaneous activity while the monkey was fixating on a blank screen. I've looked at spike phase coupling during periods of driving input, and it tends to decouple them a little bit, is what I recall. But we haven't published this work, and this is anecdotal, so don't take what I'm saying as the gospel, because I could be misremembering analyses that I've done years ago. So we have not fully investigated how these relationships change under different sensory conditions, in a very careful way. I've played around with it. In general, the rules tend to be the same from what we've seen for the visual cortex. Now, I'll take this opportunity to state where we do see a difference, and that is in looking in V1. So I've analyzed some data from V1, and in general, I find that the spike field coupling is weaker in V1, particularly in the superficial layers. But if you look in the deep in the input layers, the you find that the spike phase coupling relationship with the idea that the trough phases are depolarized and the peak phases are hyperpolarized, that doesn't do a very good job of predicting spike timing as well in the input layers. And this could be because the sources of the LFP we remember are on electrodes aren't the same sources that are contributing to the spiking activity in the input layers. So there could be laminar differences in these relationships. There could be areal differences in these relationships. And there could be sensory condition differences in these relationships. So without doing a very careful exploration of all that, I'll just say that the relationships that we see here in this data are limited to periods of fixation of a blank screen in area MT, but we see similar patterns in other areas. Yeah.
Thank you. And well, second question is about modeling the traveling waves. So far you have used integration and FAIR model, but it's not as analytically trackable as some maybe like weakly coupling models. Have you considered those alternatives?
Yeah, so Lyle's group has been exploring these types of wave dynamics and other types of models, mean field models, oscillatory coupled models, and they've been showing that the principles at play are still at play. It's just in those models, they're using these sort of surrogate mechanisms to work as a proxy for this underlying synaptic architecture. So I think that they're entirely consistent with the mechanisms that are underlying the models that I presented here, but allows them to, as you stated, in a more sort of analytically tractable way, explore other kinds of questions and other kinds of mechanisms. So yeah, I think the best solution is to use the model that's going to allow you to test the question that is at hand. And here we were interested in testing how the synaptic activity in the horizontal fiber network could explain the phenomenon that we were seeing. And so we wanted to use a model that had those as the underlying sort of sources of fluctuation and variability. But yeah, I mean, I think other models could be useful for testing other theories or other hypotheses, and I'm in favor of it.
Okay, thank you very much. That's all my questions.
Yeah, thank you so much.
Very cool. Thank you. We are running a little bit over, so we'll just take one more question. This next question is from AJ Simon.
I did not think you were going to get to my question. Thank you. And thanks for the great talk. Yeah, so I'm just curious if you've thought about retaining any information from frequency transitions. It seems like one of the benefits from your wideband approach is it almost becomes like agnostic to frequency, so it can improve phase coherence detection, but like there might be something relevant in like the transition from a lower frequency to a higher frequency during perceptual processing, for instance, that you might be missing with that. So yeah, I'm just curious if you've thought about that, or yeah, what?
Yeah, it's a really good question, and you've hit on the head with the limitation is that if you are operating in a framework, let's say where I'm interested in the frequency content of the signal, and I know that attention reduces power in alpha frequencies, or or whatever working model you have in your mind. And I want to use that as an index for whether or not the monkey is attending here or attending away, or I can imagine a framework in which you want to have that kind of frequency resolution to be able to index something. And if that's the case, then yeah, you run the risk of missing out on being sensitive to those types of manipulations. And so in which case, a different strategy for processing the data might be appropriate for addressing that question. But the concern I would have is making sure that by taking that approach, you're not also doing the being sensitive to or careful of the distortions that can occur in your signal when you are not allowing it to move in frequency as it is moving. So if I have a signal that's at 8 hertz and it's moving to 10 hertz, my 7 to 9 hertz filter will be doing a really good job on its 8 hertz, but as it starts to move to 10 hertz, I'm going to start to be misrepresenting what that signal is. And so coming up with a strategy for doing that could be done. I don't think that means that you shouldn't consider things in a narrowband state. It just means that you need to be careful to make sure you're not corrupting your signal by using a filter that is not well suited to what the instantaneous frequency content of that signal is. But yeah, so I mean, if we wanted to start asking questions that were more frequency dependent, we would have to come up with new tools to validate that we're still recovering things, and using this measure of spike field coupling as an indicator, because you can test empirically. You can say, okay, well, if there really is a signal in my, that's embedded in noise, filtering should uncover it. And if that's the case, then whatever gives me the best, whatever best recapitulates that signal is going to be the best set of tools for doing that. And anything that produces a weaker signal to that would be an indication that you're missing out on something, you're leaving something on the table. And so my view is really just test the priors and test the settings of our analyses to check to make sure that we're not missing out on something or imposing a structure that is not necessarily there in the data.
Cool. Thank you.
I think, hey, Uma, I think we should probably wrap up because it's been going a while now. But these are really good questions, and these are kind of my favorite kind. Zach, you give a great talk, so thank you so much. And these are the questions are the kinds of questions that open like new directions of research, which is makes me excited about this whole. I feel like we're onto something important here in the brain with traveling waves, and I think your talk exemplified that in the Q&A, especially. So, so this made me. So thanks a lot.
Yeah, it was wonderful. Thanks to everyone for their questions, and thanks to everyone for attending. And I'm happy also if anyone wants to follow up on anything, feel free to contact me and we can discuss. But yeah, I look forward to sharing our work going forward, and I look forward to seeing everyone else's work as they contribute to this field.
Zach, yeah, I'm sorry we ran out of time for all the questions. Did you have your email address on the screen? People know how to get in touch with you?
I don't. Right now, both my Salk and Utah emails are up, but it's just zdavis@salk.edu or zac.davis@hsc.utah.edu. But if you go to the faculty page for either the neuroscience group or the Moran Eye Center, you can dig me out and you should see my contact info there.
Sounds great. Oh, there we go. Thank you, Irfan. All right.
Perfect. Thank you.
All right. Well, thank you again, Zach. We're going to stop now. If anybody is curious for more of the information, we're going to record it. This talk is recorded. We're going to put it on YouTube sometime soon. Have a great day, everybody. Thanks for your support of this Wave Club. This is really great. Take care. Zach, thanks again.
Thank you so much. Bye-bye.